Welcome to the lecture on forensic DNA. At the end of the lecture, you will be able to explain on DNA and DNA profiling, list the samples used in forensic DNA studies, describe the common methods used in forensic DNA analysis, explain the use of DNA in criminal trials. What is DNA? DNA, or deoxyribonucleic acid, is the hereditary material in living form. It presents in every cell. In human, animal, DNA can be categorized into two types: genomic DNA and mitochondrial DNA. The important features of DNA. Uniqueness. Every human being have different DNA profile, even though 99% of our DNA remains same, except for identical Siamese twins. DNA being passed down from father and mother. DNA profiling, also called DNA fingerprinting, is the process of determining an individual's DNA characteristics, which are as unique as fingerprints. DNA analysis is a most powerful tool for human identification and has clear forensic applications in identity testing, crime scene and mass disaster investigations, and parentage determination. Blood is the most common sample used in forensic DNA study. Buccal swab and bone are second choice. Bone can be used for identification of murder or missing person. Scalp hair, armpit hair, and pubic hair can be used. Genomic DNA is at the root of the hair. Sperm and vaginal lubrication for rape case identification. They can be obtained from clothing and swab from private part, vaginal and penis. Sperm can survive in the foreign for 3 days, 72 hours. Fingernail clippings can be used in murder or rape case. Saliva and sweat can be used in shooting, drug smuggling, robbery, burglary, and break-in cases. PCR analysis is a technique that allows technicians to create millions of precise DNA replications from a single sample of DNA. In a PCR reaction, primers determines the region of DNA that will be copied or amplified. Primers are short pieces of single-stranded DNA, usually around 20 nucleotides in length. When the primers are bound to the template, they can be extended by the polymerase, and the region that lies between them will get copied. This cycle repeats 25 to 35 times in a typical PCR reaction, which generally takes 2 to 4 hours, depending on the length of the DNA region being copied. If the reaction is efficient, works well, the target region can go from just one or a few copies to billions. The results of a PCR reaction are usually visualized, made visible, using gel electrophoresis. In RFLP, the different lengths of DNA fragments are analyzed. Large sample sizes are needed for RFLP. These fragments are from the digestion of a sample of DNA with a restriction endonuclease enzyme. The enzyme chops DNA in a certain style, the restriction endonuclease recognition site. Whether or not particular recognition sites are present will provide different lengths of DNA fragments, which are then divided up through electrophoresis. Examples of genomics location used for DNA analysis. Short tandem repeat or microsatellite is a tract of repetitive DNA in which certain DNA motifs, ranging in length from 1 to 6 or more base pairs, are repeated, typically 5 to 50 times. Variable number tandem repeat or metasatellite is a tract of repetitive DNA in which certain DNA motifs, ranging in length from 10 to 60 base pairs, are typically repeated 5 to 50 times. Single nucleotide polymorphisms is single base differences in the sequence of a DNA. Mitochondrial DNA is maternally inherited and very valuable in investigations for a missing person because nuclear DNA might not be present in old samples. Y chromosome is paternally inherited and aid in identifying familial ties in males. One, a cell sample is taken, usually a cheek swab or blood test. 2, DNA is extracted from sample. 3, cleavage of DNA by restriction enzyme. The DNA is broken into small fragments. 4, small fragments are amplified by the polymerase chain reaction, results in many more fragments. 5, DNA fragments are separated by electrophoresis. 6, the fragments are transferred to an Agar plate. 7, on the Agar plate, specific DNA fragments are bound to a radioactive DNA probe. 8, the Agar plate is washed free of excess probe. 9, an X-ray film is used to detect a radioactive pattern. 10, the DNA is compared to other DNA samples. Application of DNA evidence in criminal trials. Assist in criminal investigation: murder, rape, break-in, drug smuggling, possession of firearm. Assist in paternity case to determine the biological father, mother, and child. Rape, baby swap, bogus identification. Human remain identification. Arson, fire case. Accident, drowning. Disaster victim identification, DVI. Earthquake, flight crash, helicopter crash. Thank you for your attention.